Hello, welcome to Biological Psychology. In this video, we're going to address biological psychology at a micro level. Specifically, we're going to emphasize cells. This is to be contrasted with an earlier video where we addressed biological psychology at a macro level, and also other videos on the micro level that might address such issues as cell-to-cell communication or neurotransmitters. Let's begin with a historical approach and try to understand how people through time have thought about the brain and the cells within the brain. The earliest known symbol for brain appeared in Egyptian hieroglyphics about 1700 years before the Common Era, and here from Wiki we have that symbol. So the ancient Egyptians thought the brain was important enough to create a symbol for it. Moving forward in our timeline, and also moving a little bit to the north, geographically out of Egypt, we can come to the era of the ancient Greeks. And here we see a divergence of opinion about the brain. One of Pythagoras' students believed the brain to be the basis for the mind, and that's a view that is contemporarily held. Likewise, most contemporary researchers hold the view that the brain is the seat of intelligence. And these are some ideas that had been expressed even in the ancient world of the Greeks. Interestingly, not all of the ancient Greeks held that view. Aristotle himself, one of the greatest thinkers from that era, believed the heart was the seat of intelligence rather than the brain as the seat of intelligence. And his line of thinking was that the brain might cool the blood and that humans would be more rational than non-humans because we had larger brains, therefore we could cool more blood and we could remain relatively more rational and a little bit less emotional. So you can see that even within the ancient Greeks, there was a divergence of opinion on what the brain was doing. If we move forward again in our timeline, one more time, and a little bit west and a tad to the north, we can wind up in France in the very late 1500s and look at the work of Rene Descartes. Like Aristotle, Rene Descartes was a biologist of sorts. He was also a philosopher, just like Aristotle was. Rene Descartes had performed many dissections of the brain. And in doing so, he found that generally speaking, there was a left and right component to many of the structures in the brain. But he found an exception to that general pattern. Specifically, based on his analysis, he found no lateral symmetries for the pineal gland. That is to say, he didn't find a left structure and a right structure. And this was, in his way of thinking, relatively unique. So he wasn't sure what to do with that. He began to make an attribution that maybe the pineal gland is the body component that has a soul component somewhere else, perhaps in some other dimension. So he would assert that the pineal gland had been the seat of the soul. That's not a view that's contemporarily held, but based on the information that he had at the time, that was his speculation. By the way, we now have advanced instruments that can show the pineal gland actually does have a left and right component that are largely bilaterally symmetric, but this was not available to him with respect to the technology that he had at the time. If we move forward one more time now, and again to the north, a little further north in Europe, we come to Germany in the late 1700s, and we come to an idea from Gall, which is the idea of phrenology. Here, it was alleged that different skull shapes and sizes determine thoughts, feelings, and behaviors. So the phrenologists would engage in investigating people by examining their foreheads or the backs of their heads and try to understand how the shapes and sizes of different skull types might correspond to thoughts, feelings, and behaviors. That is not a view that's held contemporarily. It's believed to be a largely non-scientific approach. It's of historical interest only. Intriguingly, and perhaps only by accident, there is evidence for considerable localization of brain functions. Let's take an example of localization of brain functions. As I've mentioned in a previous video, you'll recall that the occipital lobe, which is back here, plays an important role in vision, and the temporal lobe plays an important role in hearing. The frontal lobe plays an important role in decision-making. So there is some localization of function, but we also know that there's considerable integration. So the approach that the phrenologists had was really too crude to pass muster contemporarily, but in our review of some historical thinking about the brain, it plays an important role. Let's move forward one more time in our journey, and we see that by the 1830s, we have two researchers, who are Schleiden and Schwann, who put forth what we now accept as cell theory. And we'll go over three contemporary components to cell theory. The first is that all living organisms are composed of one or more cells. Now, some people will quibble with this and say that viruses are arguably a type of life form that happen not to be cellular. In fact, viruses are self-reproducing, and that is basically the criterion for determining whether a structure is a life form or not. But nevertheless, we can say that all living organisms are composed of one or more cells, and we can have a separate debate about viruses. Number two, the cell is the basic unit of structure and organization in organisms. So what's important here for our purposes is that we might, at various times in this course, think about a particular brain structure, perhaps the hippocampus, which plays an important role in memory and also in spatial navigation. The hippocampus will consist of many, many cells, but the cell will be the basic unit of the organization, even for that particular structure. Finally, we can say that cells arise from other cells. Our purpose here today is not to describe cellular division, but we note it as an important component of cell theory. Cell theory gives rise to the neuron doctrine. The neuron doctrine holds that the nervous system is made up of discrete individual cells, and this discovery is coming to us by Santiago Ramon y Cajal. And he made some important drawings after looking through a microscope at different kinds of cells from the pigeon cerebellum. You might recall the cerebellum from an earlier video. Here we have these drawings, and the first one shown as letter A is a Purkinje cell, and we have a slightly different cell variety, a granule cell. But these are hand drawings, and as impressive as they are, I find them very impressive, I hope you do too, they don't quite match the grandeur that we would have of an actual photograph. And here's a more contemporary picture taken at 40 times magnification. And this comes from a staining process. There are many different kinds of staining processes. This is called a Golgi stain, and it's a pyramidal neuron, again from the hippocampus, this time from a patient who's suffering from epilepsy. Okay, so with that as background, we're going to try to define what a neuron is. It's actually a very specific kind of cell, and we can define it this way. An electrically excitable cell in the nervous system. That's the most important part here in this definition. It's an excitable cell in the nervous system that transmits information through electrical and chemical signals. Now, in a future video, we'll be understanding more fully that the communication and the information that's being sent within a cell tends to be sent electrically, and between cells it's sent chemically. So that's electrical information within the cell, chemical information between cells, but we'll say more about that in the future. Here we want to emphasize the relationship between a neuron and the broader variety of cells. So here we have a Venn diagram. And the Venn diagram is indicating that within the circle we have all possible cell types, if you will, in this simple cartoon. Within that collection of all possible cell types, there's one very special cell type called the neuron. All neurons are cells, but not all cells are neurons. This shows the relationship between neurons and the wider body of possible cell types. Please do not confuse this diagram with an actual drawing of cell types. That is to say that a neuron is not a structure that exists inside of another cell. A neuron is a cell. There are other types of cells. This is simply a Venn diagram showing the relationship between this very special type of cell. Let's move on and try to understand the components of a particular neuron. So now that we know that a neuron is a special kind of a cell, let's understand its components. On a broad scale, we can say that the neuron has three major components. One is a dendrite, another is a nucleus, another is an axon. My little mnemonic for remembering these three, and in this order, would be DNA. This is not to be confused with deoxyribonucleic acid, but it just helps us as a memory trick. DNA, dendrite, nucleus, and axon. Let's take a look at a couple of diagrams, because sometimes a picture will say 1,000 words. Here we have a neuron, and the dendrites are over here. These are extending away from the cell body, which contains a nucleus. Okay, so here's the cell body, also called the soma. It contains the nucleus, and extending away from the cell body are different kinds of so-called processes. Most of them are dendrites. And they're here, and they look almost tree-like in their structures. One particular process is called the axon. It's coming out of the back here. It is unusually long, and there's only one, and so we have dendrite, nucleus, and axon. We will zoom in future pictures and learn a little bit about the node of Ranvier, the myelin sheath, a particular kind of helper cell called the Schwann cell. What we want to note for here, and for our purposes now, would be that we have a dendrite and a cell body or nucleus shown here for the nucleus and the axon coming out here, the axons eventually terminate, often before the onset of another neuron. So here's the same picture copied over again, and the space between these neurons, dendrites over here and axons over here, will be called the synapse. Okay, let's move a little further along and try to understand something else about the cells that are neurons. Many of the neurons have myelin. Myelin is a fatty, whitish sheath around the axons. And this is important because the myelin speeds neuronal communication. That is, as we have a signal that's being sent from here down this one axon, these myelin sheaths actually help to insulate the axon, and by insulating the axon, that electrical pulse moves down the axon faster. So this neuron can speak more quickly to that neuron because information is moving from here to the axon terminal more quickly. So this is an important component of the myelin. Additionally, myelin production begins about in the 14th week prenatally. So when young babies are born, they will have some myelin, but not very much. Myelin is actually generated throughout your early years and even into early adulthood. I think fascinatingly, the frontal cortex, an important portion of the frontal lobe, has myelination that is continuing up until about age 25, at least approximately. So many college students have frontal cortices that are not fully myelinated. Okay, let's learn about glial cells. We can think of these as helper cells. These are non-neuronal cells that, if you will, protect and serve neurons. And I'll dial back just for a moment to see that we have a Schwann cell as a type of helper cell, and the Schwann cells will be providing the myelin that's so important for moving the electrical information down the axon. Okay, so now let's go to understand different kinds of glial cells. We have one variety called the oligodendrocyte. Can you say that with me? Oligodendrocyte? Okay, there you go. And these supply myelin in the central nervous system, okay, which is one of two portions of our nervous system. You'll remember from another video, we also have the peripheral nervous system, and we have here Schwann cells that are supplying the myelin in the peripheral nervous system. So these two kinds of glial cells provide similar kinds of services, but they're actually distinct cell types. One occurs within the central nervous system, the other within the peripheral nervous system. So let's take a look again at a diagram. This appears to be the axon in this reddish maroon color, and it's being surrounded by some myelin that would be coming to us from an oligodendrocyte in the central nervous system. Notice that the oligodendrocyte has its own nucleus, so it's its own cell type. It's helping out this neuron by insulating its axon so that the electrical potentials can move down the axon in a domino-like manner even faster. In between these myelinated areas, there are these very, very small gaps called the nodes of Ranvier, and we'll see in a future video that there are important structures that occur at the node of Ranvier. But for right now, we'll just proceed into a very complicated picture of the neuron. And this is far more complicated than we're going to need, at least with respect to all the little details, especially when we unpack the nucleus. The nucleus is absolutely fascinating, but beyond the scope of our conversation today. What we want to focus on instead is that this maroon-looking neuron has dendrites up here, and then we also have the cell body. Toward the end of the cell body, we have the axon hillock, and this is the juncture between the axon and the cell body, and this is where the action potential that we'll learn about begins. We basically have a decision, if you will, that's made here about whether to fire an electrical signal down this path, and that signal is aided and sped up by these myelin sheath components along the axon. And it's very interesting that the signal can travel down this axon at varying rates. For example, we can have the information traveling down that axon as slow as 1 mile per hour or as fast as 250 miles per hour. So that's quite a range, and most of that variability in speed is driven by how well myelinated or insulated that particular axon is. We call that speed, by the way, the conduction velocity. That's the speed at which the action potential will move down the axon of a neuron. Okay, let's go a little bit further and understand that we have both white matter and gray matter in the brain. White matter can be thought of as a region of the central nervous system containing myelinated nerve fibers. We can think of this by way of a computer metaphor. The white matter would correspond to the connection speed of your computer. If you've been on a computer and you've tried to connect to the internet, no matter how fast your own computer is, it might be that you have a slow internet connection speed. Well, this is roughly analogous to the speed that arises from the white matter. Again, that's the myelin sheath. We can contrast that with the gray matter. That would be the region of the central nervous system containing cell bodies and dendrites and non-myelinated axons. For example, that little component that we saw called the node of Ranvier is not myelinated, and that would appear gray to the naked eye. Informally, the gray matter reflects the computer power. It's actually the gray matter and the axon hillock which will do the deciding about whether to send an action potential or not. Okay, we'll conclude with this glorious diagram of an actual brain, and what's really great here is that even to the naked eye, we can see a portion of this diagram looking relatively white and others looking grayer. So this would be myelin and non-myelin inside of the brain. Thanks for watching.